And that was really a great rundown of all of the clinical trials, both past and present. And that is something that we get a lot of questions about from patients. Where are their clinical trials? Who is to be included in these clinical trials? How do I find out about them? And what we always recommend is, first of all, the websites and the resources that you provided for us. I think those are great to scan. And above all, talk with your doctor and talk with your treatment team, because many of these clinical trials have very specific inclusion and exclusion criteria. And the great thing is to be aware of them and ask the right questions to see if it is something which is appropriate for you. So I'm going to talk just for the next couple of minutes about some of the things that happen before you get to a clinical trial. So when the doctors talked about a clinical trial that is either testing a specific new drug or comparing drugs or trying to figure out the safety profile of a medication, there are years and years and years of work before you get to that point. And a lot of the work that even leads up to the preparation of a clinical trial is some of the work that we fund at NETRF. So I thought it would be great just to take a couple of minutes, tell you a little bit about our approach to funding research and some of the really exciting projects that we have ongoing right now. And then really how you can be involved in some other areas of research. So really what we've been doing since 2005, and our mission is to transform the research landscape. We talked this morning about the fact that there's not a lot of government funding for neuroendocrine tumors, and that's where we kind of step in, try to fill that gap. So if we can accelerate progress, if we can build a community of researchers that will convene and collaborate, then that is all to the better, because then we have everyone working together, not only nationally but internationally, to solve some of these very important questions that then will lead us to maybe some targets for drugs and some other improvements in treatment. So in addition to all the work that we support to do what we call cracking the code, we also want to create that collaborative scientific community. One of our scientific advisors will always say that in order to treat these tumors, we really have to understand them. And because we have not one disease, but a collection of different diseases, we don't understand everything that we need to understand today. So that's why the work that we do to convene this community is ultimately very important. We also want to make sure that we're leveraging the scientific discoveries that are being made in other areas and see what techniques, what ideas will apply to NETs. How can we bring in some new ideas to spur along that acceleration of progress? And one of the other things that characterizes neuroendocrine tumors is that we don't have reliable cell lines. We don't have cells that will just continue to grow and multiply very, very quickly that is actually a resource for researchers. So we don't have that. These tumors grow very slowly in many cases. They will not replicate. So we've been actively trying to invest in trying to bring forth some cell lines that will be good resources for research. And we've been very successful. You can see that we have been able to put almost $19 million into net research since we began in 2005. And you see the large hockey stick line there at about 2016. We were eternally grateful for a wonderful gift from the Margie and Robert E. Peterson Foundation that has really helped us to accelerate the growth. And now really our challenge is to sustain that and to broaden our community so we can keep up this acceleration of research. We have a very specific process, and I think it's important for everyone to know that when we get applications for research, we have a board of scientific advisors, many of whom have served with us since we began in 2005. And they provide some of the scientific direction for our board of directors as well for all of us on staff as we go out to try and do outreach with many different researchers. These are people who are world-renowned in their fields, not all in NETs, but in other fields, and they have been very gracious to give us their time and their expertise over the past number of years. And then, as I said, they help to direct us so that we can go out and we can find the best science and the best researchers wherever they are, whether they're in this country, whether they're in Europe, where we have funded some work. And that has been very important over the past couple of years, where we've really looked for some of those innovative projects. So we've actively fostered collaboration among some of the top centers. And you'll see that when I talk a little bit about some of the projects that we currently have ongoing. So when we get an application, again, a very specific process that is used, every application is reviewed not only by our scientific advisors, but by external researchers. So if we don't have the expertise, we will bring it in because we think it's important. We think that we need to find the best science. We want to make sure that we have objective evaluation of not what will necessarily make incremental little changes in our understanding, but what will make potentially transformative advances in our understanding. So that's the direction that we get from reviewers as well as our scientific advisors, because ultimately what we want to do is we want to make very strategic investment in research and have not one project or two projects, but really have a portfolio. We have 15 projects now, and we have 30 scientists who are involved, because we don't know where the next breakthrough will come from. So we have to attack nets on all fronts, and that is very much our value, our philosophy of how we fund research. In terms of then closing the loop, once we do fund a project, and again, we want everybody to know about our process with this as well, we require progress reports of our grantees. We want to make sure that they are sharing data so that we don't have people working in silos. And we also want to make sure that those researchers present to one another. So we have a research symposium once a year. All of our grantees are required to attend, and we ask them to present in many cases, their unpublished data, so that we can get a lot of discussion going from people in other areas and other facilities to really look at what's working and how to make the best use of these projects and the resources. So as we attack on all fronts, we have many different types of grants. We have from the collaborative grants that we do through other organizations. We have pilot grants if we have a younger researcher who brings us a great idea. It's a kind of a proof of concept, so there's a little money to get them started on their path. We fund investigators specifically. So if we have someone from another field and they want to devote themselves to NET, then we help support them for a year or two and have them make the commitment to this field of study for their career. And then we have our largest grants, our accelerators. They're 4 years. They're the largest scope, and we started giving those out actually this year. To really sustain those teams and those very high-level collaborations where we think there is the potential for transformation. So the Accelerator Grants, we have 3 teams in place right now. The first one is looking at what causes small intestinal nets. What are the possible causes of mutations? Are they environmental? Are they infectious? Are they genetic, either at birth or acquired? And this is a really interesting team that I think came about because they were researchers who were talking at one of our symposium sessions. And we have Dr. Meyerson from Dana-Farber and Harvard. We have Dr. Nakakura, who's a surgeon in California. And we have Dr. Thirlwell, who is a genetics expert in London. So the 3 of them have come together, bringing their individual areas of expertise to really look at something from many different viewpoints. We also have a team in place that's looking at the original neuroendocrine cell. What happens to the cell of origin, and why does it start to behave differently? And what do we need to learn and understand about that process, hopefully to be able to halt it or to look at ways that we can slow it or impact it. And then we mentioned the lack of a cell line. We have Dr. Hans Cleaver, who is world-renowned in his study of organoids, which is really growing mini organs in a dish. And he's looking at how can we create these organoids and use them in lieu of a cell line to test treatments and to look at interaction with drugs. So that's a really exciting project. But I think what I'd like to do, rather than me try to tell you a lot more about their project, is really just show you a very short video, and they'll talk about it in their own words.
first round of Accelerator Grants we've chosen to dedicate to understanding the genetic and the epigenetic mechanisms that underlie the disease.
The main aim of our grant, in a nutshell, is we are trying to discover genes and pathways that made the normal enteroendocrine cells that's hiding in the gastrointestinal tract to become a carcinoid tumor and provide druggable targets that we can manipulate and hopefully find a therapeutic agents or even cure for these tumors.
We want to be able to take small bits of the tumor of individual NET patients, expand them in the lab, create large biobanks, so in the end we hope to have hundreds of samples from hundreds of patients, capture the diversity of the disease. Every patient is unique, and we can, for instance, use these banks to find out what the best drug is for each individual patient.
So what we're doing now is try to figure out what are the causes of small intestinal nerve tumors using this multifocal sample set as the study set. Basically what we're trying to do is figure out, are there any genetic or potentially environmental causes, such as epigenetic and or infectious causes, that are leading to the formation of these tumors in our patients.
What we could hope to provide would be possible novel diagnostics. So if we know new proteins that are involved in these processes, can we have new ways to identify these tumors and cancers and possibly new therapeutic targets.
So the potential of this project is to really validate a new diagnostic tool that we could use in the clinic setting that would be safer and non-invasive.
So the more multidisciplinary groups we have or international collaborations we have, the better the chance we have to actually get some answers to some of the major issues that exist in this area of research.
This is an investment in basic biology to understand exactly how these tumors tick.
We're hoping we're going to have a really solid foundation on which future accelerators can build in terms of more translational and especially clinical work that gets us closer to cure.
So those are just some of the wonderful people that I've personally had the pleasure to talk with, and better they explain what they're doing, because I don't like to get too far out over my skis, being a former Colorado resident. But you know, I mentioned that what's very important to us is building the bench, and these are some of the wonderful people who have now made NETs their career. And since we started in 2005, we've attracted 11 new researchers from other fields of scientific study to NETs, and they represent about 14 of our grants. And it's really so exciting to hear them talk about what they do and how they've really found a scientific home. And I think that's wonderful, and I think that's why they stay with us for so long, because they're as intrigued about the scientific puzzle as all of us are. So it really is good. And the other way that we get out to other audiences is that we do collaborative grants with other organizations that have a different or a slightly broader environment. So we work with the American Association of Cancer Research and fund a collaborative grant there. NANETS, which is an organization in North America of physicians and clinicians, so the people who see patients day in and day out, we provide a collaborative grant. And then the other organization is, it's called ERF, but it's part of the Society for Nuclear Medicine and Molecular Imaging, and I think Dr. Wall may have mentioned that. But again, we're really trying to get out into those audiences and with a lot of those researchers who could have the next breakthrough. we don't know, so we want to be as broad as we possibly can be. And then you saw Dr. Raj. Her project, she didn't talk about it in detail, but this is a great pilot project. You probably heard about the liquid biopsy and the opportunity to look for markers in blood as opposed to doing invasive procedures like biopsies. What she is doing is she's looking at not just blood, but corresponding tissue to see if the liquid biopsy will apply in nets. So think about the impact to you as patients if that, if that bears fruit, that they can do blood tests to look at progression of disease or response to different medications instead of doing biopsies. So we're very excited about that one, watching that one closely. Immunotherapy, it was mentioned this morning, that's the elephant in the room. Actually, a couple of years ago, we decided that we were going to make a pretty significant bet in immunotherapy, and right now we have 4 projects that are involved in immunotherapy. We have Dr. Tim Meyer, who's looking at the immune environment in cells. He is actually at University College in London. Dr. Halpern at MD Anderson is looking very specifically about the immune environment of PNETs and what genetic alterations are found in those cells. Dr. Kulke at Dana-Farber is looking at small intestinal tumors. Again, same thing, how do they respond to immunotherapy? How do we turn on the good immune cells and turn off the ones that may be blocking the immune system from responding? And then CAR-T, the therapy that's been approved for leukemia, which is really remarkable in that particular disease. Can we adapt CAR-T to NET? So some really interesting projects that we've been able to support. We have a lot of scientific accomplishments, some of them a little bit complicated, but a couple just to point out very quickly. We launched a tissue bank. Again, we don't have cell lines, but we launched a tissue bank back in about late 2008, early 2009 with 5 organizations because there was no repository of tissue samples that researchers could use and share. So we made an investment in that, and that led to a lot of collaborations along the way. We had a researcher, Dr. Papadopoulos, who found that DAX and Atrix genes that had never before been implicated in cancer or NETs appeared in about 40% of pancreatic tumors to be mutated. So that was really kind of seminal work that a number of our projects have now built upon to see what that influence really is and what we do about it. We also had a team that found 2 PNET cell types, kind of a type A and a type B, and those tumors react a little bit differently and may have a little bit of correlation to patient outcomes. And then I mentioned the cell lines. We've had a cell line prize that we have offered to teams that can verify that they have a replicating cell line. And for quite a long time, we had not a lot of response. Now we have a couple of cell lines that we're trying to verify. And if they are approved, then again, that would be something that researchers all across the country would be able to access. Another way that you judge scientific impact is how many peer-reviewed papers do you have coming from your projects. So we have 48 funded studies since our beginning, 48 peer-reviewed articles, not necessarily one-to-one, but coincidentally the same number. And those articles have been cited about 2,100 times. So that's another measure of success, that people are reading these articles, thinking about them, and using them to cite in their own work. So we're very proud of that. I want to finish out with just a couple of notes because we talk about clinical trials. We know that some of them are very small, some are very specific, but there are other ways that you can get involved directly in research. There's something called pattern.org, which is an organization that we collaborate with. It's actually part of the Rare Cancer Research Foundation. And they have an effort ongoing. So if, as a patient, you're having a surgery or a biopsy, sometimes when they send tissue to pathology, there's extra tissue. And what pattern.org has done is they've created a system where you can work with your doctor and also with pattern.org, and this extra tissue will be sent to pattern.org. It will be registered and sent to kind of a tissue bank to really look at how can we create a repository and start to profile some of these tumors and see what we can learn. So you can go to pattern.org. There's something in your book. It's a great organization. We feel very strongly about this, and we want to make sure patients know about that ability to really help with research, not only for themselves, but the people who will follow on. And then finally, how you can get involved with us. Again, we are just so pleased that you took the time and drove the miles in many cases to be here. We'll get you signed up for our newsletter, follow us on Facebook and Twitter, and make a gift if you want to continue to help us support the research, and just stay in touch with us. We want to answer your questions, make sure that you get what you need, and we're just very grateful for you to know about who we are, what we do, and that we are here for you.